So we talked about alginates only. And the main point about alginates is that it is a family of polysaccharides. It's not just one polysaccharide, it's a big family. And alginates are produced by brown algae for commercial production, but also, and fortunately I would say, some bacteria such as Azotobacter vinlandii and Pseudomonas aeruginosa also produce some special alginates. The seaweeds are harvested by a special vessel, special boat, having a device that simply grabs the plants from the sea, from the bottom of the sea, and takes it into the boat. The algae are then transported to a depot, and the algae are first treated with formaldehyde, otherwise they will start to decay very quickly. Microorganisms will degrade the algae if they are not preserved, and then it goes to the factory. And at the end of my talk today, I will explain how you make alginate from algae. We talked a lot about the chemical structure of alginates, and they have two components which we call M and G. And M means beta-D-mannuronic acid, this sugar. Uronic acid means having carboxyl at carbon number 6. But such a polymer like this does not exist in nature. In all cases, we have enzymes which attack the chain and pick out some of the mannuronic acid and convert into G, alpha-L-glucuronic acid. Note that even the big change of name, the chemical change is only here. The linkage and the two other hydroxyls are the same. But as we shall see, this has big consequences. Because alginates are produced by bacteria, we can make special alginates by in vitro epimerization. So in a test tube, we can make special alginate. In the case of mannuronan, only containing mannuronic acid, we can obtain from bacteria which are epimerase negative mutants. So such mutants have been made, and this material has been made in the laboratory. So it is available for research. A second extreme alginate is the one that is polyalternating, Mg, 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 almost perfect. This is obtained by using a special enzyme with this as substrate. Then we have alginates with extremely high G, maybe 90%. This you do not find in algae, but we can make from this, via this, and then use a new enzyme to make long G blocks. These special alginates have been very important to understand the function of alginate. I should also mention, I did not say this last time, but recently what we call G blocks, isolated G blocks, obtained by partial hydrolysis, such G blocks are of great interest currently, and some research projects involved in using G blocks in some biomedical applications is undergoing, and actually clinical trials for the treatment of cystic fibrosis is currently going on. I predict that the G blocks will be a hot area the next 5 years. What about in vivo, in the plant? We think that manurenan is made as an intermediate, but not as a final product. And we are quite sure that many different enzymes, epimerases, are found in the plant. So we get functional alginate, which is a heterogeneous product. And depending on the plant, depending on where in the plant, we can get different alginates with Fg varying from 0.4 up to 0.7. For instance, the industry pick the plant and split here and make alginate from leaf and from stipe. Two different processes, two different alginates. We talked a little bit about, or quite a bit about, the so-called sequence parameters. In the case of alginate, we introduce a series of parameters to describe the structure. And the method to do that was NMR. I just remind you of this without going into detail. So here comes something new. Because NMR spectroscopy was introduced in the 80s. But much of the chemistry of alginate was known before that. So how did they discover and determine the structure of alginate before having NMR? In the 1960s, some very elegant chemical experiments were done in Trondheim using heterogeneous acid hydrolysis. So they took the alginate and treated it with citric acid at pH 1.4 and of course heating. In this way the chain was broken into pieces of about 20. So dP means degree of polymerization. It is the number of sugars per chain. So they stopped it here and looked at the product. And this hydrolysate could be separated into two fractions. One fraction was insoluble, and it was termed resistant fraction because even with prolonged hydrolysis, this material did not degrade very much more. And it was found that the dP was about 10 to 20, and it contained about 50% G. At that time, they had a very slow biochemical method for M and G. I will not explain why, because nobody is using that anymore. On the other side, we got a soluble fraction, which also had a dP in this range, 10 to 20, and had about 40 to 50% G. So quite similar, except that this one is soluble and this insoluble. This turns out to be the Mg type of blocks by more analysis.